Good morning, everybody, and thank you, Ira, for your kind invitation. It's a pleasure to give this lecture for the EXTRO-Lab seminar group. I am Professor of Oncology at the University of Padova and group leader at a comprehensive cancer center based in Padua called Veneto Institute of Oncology. I'm a medical doctor with a residency in oncology, but I spent actually almost all my life in the lab. So therefore, the talk I'm going to deliver will be essentially results of lab work in the last years in my center. I'm leading a group made by 10 persons, 10 people. Most of them are biologists. And a part of our group has a special interest in a very important signaling pathway in cancer, which is Notch, and especially in the role that this pathway has in certain malignancies, in certain acute forms of leukemia. Called T acute lymphoblastic leukemia. So today's seminar will be about the dissection of molecular mechanisms of resistance to a Notch1 targeted therapy in models of this TALL. For those of you who are not familiar with this acute form of leukemia, I can tell you that TALL accounts for 15 to 25% of all acute leukemia forms in children and adults, respectively. It's a rare disease. Incidence is about 40 cases per million people per year. It has a relatively good 5-year survival rate, thanks to the effectiveness of chemotherapy, being the survival rate 75 to 85% at 5 years for children, and substantially worse for adults. For adults, it's just 40 to 60% survival at 5 years. Despite the fact that chemotherapy in general works, is effective in this disease, there remain 20-25% of sick children who cannot be currently cured with chemotherapy or standard approaches, and even more adults. For the aims of this talk, it's important to know that TALL is a prototype of Notch-driven malignancy. And this statement is supported by the fact that mutations in Notch1 or in FPW7, which is a ubiquitin ligase important for Notch degradation, are mutated in about 50 to 60% of cases of TALL. So there is a genetic link between Notch1 and TALL. Over the years, we were able to set up a mouse model of TALL. So we transferred leukemic cells from sick children to immunodeficient mice. We used the NOD-SCID mice that time. And this engraftment is quite effective. It has an efficiency of about 50% when you start from fresh bone marrow samples. So 1 out of 2 will take in mice, whereas efficiency is substantially lower if you start from frozen samples. It's, as you will see in the following slides, the model is pretty close to what one would see in patients, because TALL cells engraft in clinically relevant tissues of a mouse, such as the bone marrow, the spleen, and even the central nervous system, the brain. The other, I think, very positive feature of the model is that the blood can be used to measure engraftment and disease progression by flow cytometry, as you would do for patients. Finally, if you manage to label the T-all cells with a luciferase encoding lentiviral vector, you can image the disease, and you will see some example of this shortly. So this is basically kind of drawing of how it works. We generally start by injecting 5 to 10 million cells intravenously in the tail vein of the immunodeficient mice at day 0. There is usually a kind of 10 days window where there is nothing to measure in the blood, but starting from day 10, if you do blood drawings, you can find human leukemic cells in the blood. And these cells are easily trackable by labeling them with anti-CD5 or anti-CD7 human markers, which are expressed by the large majority of the human leukemia cells. So you can find very few human cells in the murine background. Then you can repeat blood drawing, and at the end, the mice die of leukemia, depending on the model, quite shortly after injection of the T-all cells, 17 days, 2 to 3 weeks. In some cases, you may need to wait months before the mice die. So there is, especially at the beginning, when you, the so-called primograft, when you first transfer the human cells into the mice, there can be a considerably heterogeneous time until sacrifice of the mice. At this time, we have more than 40 xenografts generated from T-all patients. Basically, all of them are children with different phenotype, risk of disease recurrence, and different response to conventional therapy patients, especially prednisone response, and genetic status in terms of notch mutations. We continue so to generate and maintain this xenograph collection is really a huge effort, because you have to periodically check the identity of the cells by running sequencing for TCR, T-cell receptor rearrangements, and also STR analysis to make sure that the cells remain what they initially were, and there is no mixed up of the xenografts over time. I spend a few words on the notch pathway, since this will be at the center of this talk. The notch pathway is one I think is well known to the majority of you. It's a very established signaling pathway. It's an intercellular signaling pathway, and it's generally made by 4 notch receptors, notch 1 to notch 4. They are expressed on many cell types, and in the case of leukemia, very important are the notch 1 and the notch 3 receptors. These receptors undergo a series of 3 proteolytic cleavages. The one is before they are brought to the cell surface, and then the second and the third cleavage happen when the Notch receptor interacts with the Notch ligand. Notch ligands are 5 and are members of the delta families, delta 1, delta 3, delta 4, or of the JEG1 and JEG2. The physiological mode of activation of Notch is through interaction with the ligand, because this interaction triggers cleavage of the S2 and S3 sites. The S3 site cleavage is mediated by gamma secretase, and as a consequence of this activation, there is the release of a domain which is called ICD, intracellular domain, or ICN, intracellular notch. And this domain enters the nuclei and interacts with CSL, MAML, and other co-activators to turn on the transcription of a set of Notch target genes, which include many genes. Some of the most well-known and established Notch target genes are S1, Deltex, c-Myc, and in T cells, the pre-T-alpha transcript. At the end, of course, Notch is also degraded, and it's degraded, the ICN, in the proteasome by polyubiquitination. In this context, the ubiquitin ligase FPW7 plays a very important role in the final degradation of the activated part of the receptor. Where are the mutations of Notch1 in T-all samples? There are some mutational hotspots, which are shown here by the triangles. Some substantial part of mutations are concentrated in the so-called heterodimerization domain, shown in green in the slide. And another part of mutations are concentrated in the so-called PEST domain, which is important for the degradation of the receptor. And as you will see in the gray box on the left, there are about 26% of T-all samples which have HD mutations only, another 12.5% which have PEST mutations only, and 17.7% of cases which have a combination of HD and PEST mutations. These two mutations have different effects in general on Notch signaling, because the HD mutations can lead to ligand-independent activation of a pathway, whereas the PEST mutations account for increased stability of the receptor. But if you have a Notch which has only a PEST mutation, that is going to be still dependent on the ligand for its activation. Of course, the most biologically powerful mutations are the combinations of the two. Because if you have both mutations in the same receptor, then you will have a receptor which is very likely ligand independent and also quite stable. So it's a receptor which is firing its signals into the cell. Not surprisingly, given the importance of Notch1 in T-all, over the years there have been many attempts to develop drugs to block Notch signaling in these cells and in general in cancer, because Notch is important also for other cancer types. The most popular and well-known inhibitors are gamma secretase inhibitors, or GSI, which block the proteolytic cleavage, the S3 proteolytic cleavage of the receptor. Unfortunately, they are not notch specific because they recognize about 90 substrates. So they block notch, but they do also much more. And their clinical exploitation has been limited both by toxicity and by, I would say, lack of strong clinical results, despite the fact that in preclinical studies they were encouraging in a way. A second strategy is represented by monoclonal antibodies against notch receptors. These are more specific because they target either Notch1 or Notch3 or one of the other notch receptors or the notch ligands. For instance, Delta-4 has been a ligand at the center of a lot of interest by the pharmaceutical industry over the years. And finally, there is a new strategy, I say, which aims to block the notch transcriptional complex by a small molecule inhibitor, which enters the nuclei and is able to block notch transcriptional activity into the nuclei. For this last approach, we have so far only preclinical data which have been published in PNAS last year. What did we do in our studies? We focused, we started these studies about 5 to 6 years ago, and we focused on an antibody which at that time was quite promising. It was a specific monoclonal antibody against Notch1 produced by Oncomed Incorporated, a US-based company. This is a human specific, binds a juxtamembrane site close to the heterodimerization domain and was shown to block the proteolytic cleavage of the receptor. There was already some information about this antibody, but it was not evaluated in primary T-hole xenografts, and that's what we did initially. We use in our experiments rituximab, which is an antibody targeting human CD20, which is not expressed in T-hole cells, as a control antibody. The initial experiments we performed were, this is in the general experimental layout, kind of experiments where we wanted to see whether this antibody was able to delay the outgrowth of leukemia in mice. So we started by injecting the T-hole cells IV, and at day 2, so at the time where the disease was microscopic, as I told you before, we injected the anti-Notch1 antibody. And these antibody injections were repeated every week. We then monitor the outgrowth of leukemia until to sacrifice of a control mice. At this time point, we evaluated blasts in bone marrow and spleen, levels of apoptosis in these organs, and then we also performed some RT-PCR studies to interrogate expression of notch target genes in these cells. What came out from these experiments? The first result was that we tried 7 different PDX models, and there was clearly a very heterogeneous type of response. These columns show the amount of CD7 positive cells in the spleen of the control mice or the mice treated with an anti-Notch1 antibody, which are in the light blue. As you can see, there are 4 different models. where there is a substantial difference between the number of human leukemia cells in the spleen of the control and the anti-Notch1 treated mice. In contrast, there are 3 models where there is almost a marginal difference. Or in one case is statistically significant, in 2 cases is not significant at all. So there are clearly good responders and poor responders. You see here also at the bottom the huge difference in the size of the spleen between a control, mice receiving the control antibody and mice receiving the anti-Notch1 antibody when it works. So this is the size also of the normal spleen, and this is massively enlarged. It's also important to stress that similar results were found in the bone marrow of the mice. Again, the same 4 PDX models were good responders, and our 3 models were poor responders. And this is how the femurs of these mice look like. As you see here, this is the reddish appearance of the femurs of the control mice, and this is the whitish appearance of the femurs of the control mice, because these femurs are completely replaced and filled by leukemia cells. It's important also to let you know that there is a marker which is which we used to detect responses to anti-Notch1 treatment in these models, and it's the expression of CD7 on the surface of the leukemia cells in the spleen or in the bone marrow. As you can see in the middle, there is the strong expression levels of CD7 in the mice which received the control antibody RTX, rituximab, whereas on the right panel there is the clearly reduced percentage and also intensity of fluorescence of CD7 on the surface of the mice treated with anti-Notch1. And the same happens also in the bone marrow. So you have a readout that you can easily assess by flow cytometry to interrogate whether your samples are or are not responding to this therapy. And this is how you can detect response by imaging, by bioluminescence imaging. This is possible only in part of a PDX because you needed to transduce them with a lentiviral vector encoding luciferase, and this is a procedure which doesn't work for every PDX, but for some PDX it works. And so here you see, for instance, in 3 different mice receiving the control antibody, how the signal starts to be seen at day 13. 2 days later it grows, and another 2 days later it's really very strong, and then you have to kill the mice. This is a very aggressive disease also in the mice, so you may have an initial delay, but when it starts growing, it grows very fast. In contrast, in the right panels, you see at the same time point imaging of leukemia in 3 different mice treated and responding to the anti-Notch1 antibody. You don't see any evidence of a disease. You start to see some dots, but clearly the mice are, so to say, clean at this time point, although of course they will eventually develop the disease. These are additional effects that you detect in these models following treatment with the anti-Notch1 antibody. One is I would say the most prominent effect is in terms of apoptosis. So anti-Notch1 antibodies in vivo induce a huge amount of apoptotic cells in the mice which respond to the treatment, in contrast, of course, to the models which do not respond to the treatment. The same is true also in the bone marrow, and you can also measure proliferation. Proliferation, as you would expect, by an effective treatment is reduced by anti-Notch1 administration. I have to stress the fact that most of these experiments can only be done in vivo, because T-all cells, which proliferate incredibly fast in the mouse microenvironment, they stop growing and die by themselves as soon as you take them and try to culture them in vitro. So it's unfortunately, I would say, very difficult to perform similar studies in cultured flasks, simply because the cells, not all of them, but in most cases, they behave very differently in the mouse environment compared to what you would see in vitro. Let me advance with the slide. There is another very important readout that you can use to monitor the effect of therapy, and it's interrogating the expression of Notch target genes. We generated a real-time panel of genes and interrogated the expression of these genes in the mouse samples treated with an antibody against Notch1 or the control antibody. And as you can see, the responder mice, in terms of if you take the level of expression in the anti-Notch1 treated mice, you see huge differences, especially for certain Notch target genes, including CR2, Deltex1, HES1, HES4, between the control mice and the anti-Notch1 treated mice. Again, if you take a poor responder, there is almost no difference in the expression level. What makes the difference between the responder and the non-responder in these systems is entirely the Notch1 mutation. So responder PDX are those which bear Notch1 mutations. In contrast, all samples which lack Notch1 mutations, I would say you may expect it, but it's turned out to be true, are completely refractory to Notch1 therapy. We then performed more difficult, I would say, experiments where we tried to detect a therapeutic effect of an anti-Notch1 therapy in a more advanced disease setting. I think this is quite an important experiment. These type of experiments are called regression trials. And I think it's important because at the end, this is closer to what you would see in the clinic. So a patient generally will come to the doctor, especially with an acute leukemia, when the acute leukemia is presents with a full-blown disease. So you need definitely at the end new drugs which work in this kind of setting. So let us see what happens if you treat the mice during detectable disease with anti-Notch1. In this model, detectable disease means day 11. At day 11, if you sacrifice the mice, you have about 5% of leukemic cells in the spleen and about more than 20% leukemic cells in the bone marrow. So it's clearly the disease is there. And you have also some leukemic cells in the blood. If you start treatment by day 11, you can then detect by the standard parameters what the therapy does in the advanced disease setting. As you see, from these columns, initially you don't measure any difference. So remember that we started treatment at day 11. So at day 14, there is no difference between the control and the anti-Notch1 treated mice, same at day 16, but you do see an effect at day 18. At day 18, there is a significant effect in terms of CD7 positive cells in the blood of these mice, and also in the spleen when you sacrifice them. Of course, in an advanced disease setting, you need to combine anti-Notch1 with other drugs to achieve a kind of meaningful gain in survival. And the drug which we selected for this combination is dexamethasone, because dexamethasone, along with prednisone, are steroids which are very important in clinical management of TOL. So the most important, I think, part of the slide is panel B, where you can see the quite important effect of a combination of dexamethasone and anti-Notch1 on the number of CD7 positive cells, so leukemic cells at different time points in the blood. And in the blue columns, panel C, you see also the effects of the combination in the organs, so in the spleen and in the bone marrow. I have to stress that even if you use a combination, there is still a not marginal number of leukemic cells in the organs. So at the end, the leukemia will very likely come back in these models, but the effect is definitely much stronger compared with what you would see if you treat the mice with dexamethasone alone or anti-Notch1 alone. So I think that also if I would transfer this to a clinical trial, I would definitely bet on the combination of the antibody with another established drug like dexamethasone and maybe also some chemotherapy drugs. Now in the final part of the lecture, I will show you what I think is to me was to me the most stimulating part of this story, namely the investigation of the patterns of resistance to this antibody in the models. As you know, maybe from our seminars or from your previous studies, in general, every target therapy ends with resistance. And also targeting Notch1 is no exception to this general rule. In the PDX models I am presenting you, we noticed different types of resistance, which are well represented in the bottom part of the slide. This slide represents 3 different PDX models: PDX 19, PDX 8, PDX 11. And you see here the survival of the mice engrafted with these different PDX cells. As you can see from the continuous lines, you have a survival which is different from model to model and ranges in this case from less than 20 days to about 30-35 days, depending on the model. When you treat the mice with an anti-Notch1 antibody as monotherapy, you have a survival gain. You have a doubling of a survival in the case of a PD19 model, so the blue dots. And you have an even stronger effect in the case of a purple model, PD-TAL11, and even more, I would say, clinically probably significant effect in the case of a green PDX model, PD-TAL8. So focus on the two extreme parts of this spectrum, the early onset resistance and the late onset resistance. So basically the focus of our studies in the last 2 years have been understanding a bit more with a kind of molecular insight the determinants of these different patterns of resistance. I have to say that we started from analyzing the gene expression profiles in the two modes of resistance, so the early onset and the late onset. In the case of the late onset of resistance, by using the same card of genes, custom panel of genes that I showed you before, we had marginal differences in expression levels of Notch target genes between the control mice and those which became resistant to anti-Notch1. In contrast, in the case of an early onset resistance, there was still a huge difference for many of the genes of the panel between the control antibody and the anti-Notch1 antibody. What is the meaning of these results? The meaning is that in the early onset resistance model, there is still, despite the fact that the mice die of leukemia, they are resistant to therapy, but the pathway, the Notch pathway, is still inhibited. In contrast, in the late onset resistance type, it seems that the Notch pathway is no more inhibited. It's quite important also to note that you have here different results when you perform serial transplantation of the resistant cells into naive mice. So if you take leukemia cells from the spleen of a mouse which became resistant to the treatment and inject them into a naive mouse and then perform treatment with an anti-Notch1 antibody, you have different outcomes of this experiment depending from if you start with a model which presents early resistance or late resistance. In fact, in the case of the early resistance, you seems that this early type of resistance is reversible. In other words, when you transfer the resistant cells to a new naive mouse and perform the treatment, you have still a response to the treatment, as you would obtain if you start with cells which have never been treated with anti-Notch1. In contrast, in the late resistant model, there seems to be a stable resistance. So if you transfer the resistant cells to the new host and perform treatment, this is a stable resistance. This is quite an interesting finding, I think. It shows you how heterogeneous can be resistance to this type of therapy. I have to say that when we made these observations, we initially tried to ask whether this type of resistance could be accounted for by known mechanisms of resistance. As I told you in the introduction, the Notch pathway has been broadly studied. There are hundreds of studies, and there are also tens of studies, I would say, on resistance mechanisms. One known mechanism of resistance to Notch inhibition is loss of PTEN. So we asked whether maybe in our models we may have loss of PTEN, but that was definitely not the case. We interrogated PTEN expression by Western blot analysis, and we had PTEN expression both in the control mice and those which became resistant to therapy in all three different models. So PTEN loss is not at play in these models. We also knew from our studies that mutations in FBW7 could be another mechanism of resistance to gamma secretase inhibitors, so to Notch inhibitors. And therefore, we asked whether we may have selected cells with FBW7 mutations in our resistance models. But it was definitely not the case. In these three models, we had wild-type FBW7 sequences. So again, resistance to the antibody was likely not due to inactivation of FBW7, which would account for increased, for reduced degradation of Notch, and so increased signaling from Notch. We then had a look, more broad view at the transcriptome of the resistant cells compared with the sensitive cells. In the case of a late onset model of resistance, there was actually patterns of gene expression were almost identical. This again suggested that in the late onset model, probably there was something happening at the level of an interaction between our therapeutic antibody and the Notch receptor. I will tell you something more about this shortly. In contrast, in the case of the early early resistance model, there were huge sets of genes which are differently expressed between the resistant cells and the control cells, which were the pathways enriched according to gene set enrichment analysis in the resistant cells, in the anti-Notch1 treated cells, resistant cells compared with the control cells. In this model, we had enrichment of Notch signaling. And it's different between the two groups, as I told you already. But interestingly enough, there were also many pathways differently expressed and related to cholesterol homeostasis, adipogenesis, and fatty acid metabolism. So there was here, according to the gene expression profile data, a metabolic rewiring associated with this early type of resistance to Notch. The gene array data were in part confirmed by quantitative PCR of some of these metabolic genes, which are shown in this slide. And as you can see, the resistant cells, which are shown here by the blue dots in this image, have invariably much lower level expression levels of these transcripts compared with the control cells. And these transcripts all belong to the mevalonate pathway, to the pathway which leads to the synthesis of cholesterol in the cells. And the fourth transcript that I have shown you in the slide before, ABCA1, is a major regulator of cholesterol transport in the cell. So there was really, from these data, we tried to, we had the feeling that the early type of resistance to Notch inhibition had to do with rewiring of fatty acid and cholesterol metabolism. And therefore, we went on and performed metabolomic analysis of the leukemic cells in this model. We interrogated kind of more than 300 metabolites, and the results of this metabolomic analysis was that there were clearly many metabolites less abundant in the anti-Notch1 resistant cells compared with the control cells, and they are shown in red in this picture. And most of these metabolites belong to these lipid classes. So oxysterols, glycerophospholipids, glycerides, ceramides, sphingomyelins. There are also many other metabolites which are not related to lipids and are not shown here, which were, however, not statistically significant, different between the two groups compares. So I think here we have a kind of confirmation at a different level, at the metabolomic level, of what we have seen at the transcriptomic level. Lipids, of course, and cholesterol is a very key component of the cellular membranes. It can reduce the membrane fluidity. It's a component of lipid rafts that are important for endocytosis. It's important for anchoring of proteins to membrane, and it's a precursor of different molecules. So it's clearly a lipid which has a key biological role in all cells. Cholesterol can be synthesized starting from acetyl-CoA or can be taken up from the circulation. And there are also some forms of storage of cholesterol and other fatty acids, triglycerides, in the cells, which are lipid droplets. So we also measured the amount of lipid droplets in our leukemia cells by labeling lipid droplets with body pi 493-503, which is a fluorescent dye. And so you can analyze the amount of lipid droplets by flow cytometry. And as shown in this picture, especially in the B panel, there was a significant reduction in lipid droplets in the anti-Notch1 resistant leukemia cells compared with the control cells. Again, somehow confirming that there was quite a strong metabolic rewiring in these cells. We also noticed that in these cells, the amount of Notch1 full-length protein expressed by the resistant cells was clearly lower compared with the control cells. You see in this Western blot analysis 5 different replicates, so 5 different mice samples from control and anti-Notch1 resistant mice. And there is clearly a reduction also of Notch1 full length. We speculate that the metabolic rewiring that I described to you may be somehow involved in the different abundance of Notch1 in these 2 sets of samples. So this is, we are close to the end of the seminar. The working hypothesis behind the early onset of resistance. We think that there is a central modulation of sterol synthesis, and we argue that this may modify the membrane composition and or its fluidity, which again could account for variations in endocytosis and vesicle trafficking, and these could at the end account for the reduction of the expression level of a cell surface CD7 and possibly of Notch1 expression. The possibility is, of course, that the disease becomes, you have less Notch signaling, but there is a kind of residual disease which continues to be there and to proliferate, although at a lower rate compared with the parental cells, and that eventually accounts for resistance. I spend also a few words on the other model that I showed you before, so the late onset of resistance, the model where you have resistance after more than 160 days. In this case, we suspected, for the reasons I told you before, a genetic basis for that form of resistance. And so we performed analysis of chromosomal aberrations in these samples, as well as mutations. From the viewpoint of chromosomal aberrations, we did detect some changes compared to the normal, let's say, structure of the chromosomes, but there was no resistance-associated chromosomal rearrangement in these samples. In contrast, when we performed whole exome analysis of the resistant cells compared with the control cells, we found many, of course, variants, but with regard specifically to Notch1, we found that the resistant samples had 2 Notch1 mutations, which are always the same, and were also confirmed by Sanger sequencing. And these mutations were in a portion of the Notch receptor, which is important for antibody binding. These are basically the conclusions of this part of the study. We highlighted the existence of 3 different types of resistance to Notch in terms of time of development. I spent very few words on the intermediate onset. We have also much less data about that. Second parameter is the stability of the resistance, which is stable in the case of a late onset and transient in the case of an early onset. Differences in terms of Notch signaling, differences in terms of transcriptional levels and of genomic alterations. So the take-home message from these studies is the extreme heterogeneity in the molecular features of resistance to anti-Notch1 antibody. And the fact that these mechanisms are completely different from those which were already described and associated with resistant to gamma secretase inhibitor. The model that I like and I think can also apply to these experiments has to do in general with our view of drug resistance in cancer, and we simply apply it to the PDX models. You can have intrinsic drug resistance, shown in the top of the panel. In the experiments I discussed with you, that's the case of the T-all samples which lack Notch1 mutations. These samples have a kind of intrinsic drug resistance. The target is there, but it's not activated Notch. So there is really not much room for a target therapy in this, for a Notch target therapy in this context. More common is the case, or more intriguing, is the case of acquired drug resistance, and you can have at least two types of resistance. The selection of drug-resistant clones, shown by the blue cells, that's something that I think we found in the late onset resistance model. But you have also this kind of, I think, epigenetic form of resistance shown in the bottom by the gray cells. So there are cells which are drug tolerant or persister cells. And then probably with time, within these persister cells, there happen to be additional, maybe genetic changes which at the end convert these cells into kind of stable resistance. I had not time to discuss it with you, but this is exactly what we had in our model. So if we perform treatment of the leukemia cells which have this kind of reversible resistance, 5 to 6 times in different hosts, so a serial treatment, at the end we end up with a kind of stable form of resistance, which is, I think, quite in line with the predictions from this model. Finally, if somebody from the audience wants to have more details about what I told you, especially in the second part of the lecture, you may have a view of our hematologica paper published last year, exactly on this topic. And this is the long list of our papers on the PDX models. It's just to let you know that, as told you, it was a huge effort to establish this PDX collection. But once you have it, it's clearly a resource for the laboratory because it's open to your own studies as well as to international collaborations that we are very lucky and honored to participate over the years. Finally, I would like to thank my historical, I would say, consolidated collaborators on these studies. Sonia Minuzzo first, who was actually the person who established the PDX and still takes care of them. And then she was flanked by Valentina Marica and Angela Gassi for the bioinformatic analysis. We had also a very fruitful collaboration over the years with the Department of Pediatrics. We were close by, and this was key to generating the PDX without the need of freezing them. And I also thank Adolfo Ferrando for his suggestions and collaborations, part of his studies. The Oncomed for providing us with the antibody and tips about how to use it. And our sponsors, so Italian Association for Cancer Research and the Cariparo Foundation, which founded me for 3 years along this team. And I thank you all for listening to this lecture and open to any questions.
Thank you very much, Stefano. I have received already the request to state the question. Edith, are you here?
Yes, I'm here.
Okay, hi.
Hi, how are you? Hi, Stefano, thank you. It was very interesting. I wanted to ask you about the resistance and the cholesterol pathway. The cholesterol pathway is also leading to NF-kappa B activation. And I was wondering whether you can see any role for inflammation in this system, because, and this mainly connects to the effects that you also see with dexamethasone that can also act through being anti-inflammatory. And the reason that I'm asking is that we have a publication from last year in which we have shown that NF-kappa B activation activates Notch1. So, so this might not apply to the early resistance, but overall, maybe there is an involvement of inflammatory mediators in all of this resistance process.
Yeah, it's a good question, a good point. I think that in the past there have been also other papers indicating a link between FKB and Notch1. I think that Yannis Aifantis, New York, published a couple of papers on this topic in the past. And it's quite likely that an FKB plays a role in this system. I have not directly investigated that, and maybe there could be some room for investigating this, because it's very reasonable that an FKB modulation may contribute to this form of resistance. Yeah, it's quite possible.
Okay, thank you.
Thank you, Adit. Does anybody have a question?
Hi, this is Barry from Cologne. So, Stefano, thank you very much for your very nice talk. I think it's very successful for you to show the experience of the PDX model concerning of the DAL. I want to ask concerning of the immune modulation influence for the real patient. Have you ever to think about to use the humanize the PDX model in the future, for instance?
Yeah, very good question. Indeed, the limitation of well, these PDX have some advantages. For sure, they are much more closer to patients than, I would say, T-whole cell line injected into a mouse. But the microenvironment is, and especially the immune, the lack of immune cells could be a weakness point of these PDX models. At the time we started these experiments, I think there were not yet so well-established models of humanized mice, which are nowadays more reliable and set up. So I think if one should start these experiments now, that would be make a lot of sense to use the humanized mice. Although it's, of course, it adds a level of complexity to the system. So it's there is, of course, another possibility that it could be that especially T-cell responses might influence the activity of anti-Notch1 antibodies. And on the other end, it could be the anti-Notch1 antibody has some effect on T cells, because also normal T cells express some levels of Notch1. So I think that if we could really work with humanized mice, that would be, we would be a step closer to the patients. But it's not easy to set up. That's something you have to know. Yeah.
Mm-hmm. Yeah, and I also saw the data what you show is like for the fresh samples you have like 50% of success rate, and for the frozen samples, right, you have like 10% rate. So for those frozen samples, could you share the experience? Do you have some limit, for example, how many years or how many months of this foreign?
Well, first of all, I think that speaking of fresh cells, the percentage of engraftment can be even higher. I say 70% if you choose NSG mice in place of not SCID. But of course, our colony was established 5, 6 years or 7 years ago, and at that time the standard was not SCID. So you can gain a bit if you use even more immunodeficient mice. That's in general.
Mm-hmm.
Second, with regard to the frozen cells, for unknown reasons, there is a huge variability in the recovery of cells, of frozen cells. So despite the fact that you follow the same protocol every time, some some patient cells suffer a lot from freezing. We have improved this a bit by using commercial freezing medium.
Mm-hmm.
We use CryoStor CS10, and that's better than the standard FCS DMSO that most of us, including ourselves, used to do in the past. But still, there are huge differences in the survival of the frozen cells depending on the donor. And this is something you can't predict.
Okay.
I tell you, we once the PDX is engrafted, then it's you can rely in general on frozen cells from the PDX. But the very critical point is the patient's cells.
Okay.
So the faster you are in injecting the cells into the mouse, the better it is. For instance, in the past, we have provided Columbia University with several of these models. And one may wonder, it could be a bit strange that a small laboratory provides such a big university with its reagents. And the reason was that due to their organization, was very complex to inoculate very rapidly the cells, the patient's cells into the mice. There were always some steps in between, and time was passing. So the key point in being successful is to be as fast as possible from the patient to the mouse and possibly avoid freezing. If you have to freeze them, then choose a commercial product.
Okay. Thank you very much for sharing this very precious experience. We learn a lot. Maybe we send you email later on if we have further questions. Yeah, thank you.
Welcome.
Okay, Stefano, you have some comments in the chat. If anybody has the questions, please don't hesitate to state them, and Stefano will be happy to answer all of them. Are there any further questions? Yeah, I don't see any questions in the chat, and nobody asks using their micros. Therefore, I would like to thank Stefano for excellent lecture, and we will have another lecture from Stefano, but it will be scheduled either in summer or in autumn. You know that we have the seminars every 2 weeks, and next seminar will be after 2 weeks, and you will be informed as usual. Thank you very much for your participation, and see you later in 2 weeks. Bye.
So have a nice day.
Thank
Schönen
you.
Tag.
Same, yeah.
Auf Wiedersehen.
Thanks.
Bye bye.
Bye.
Thank you.